Hello everyone, I hope you had a good break. So, continuing, I am going ahead with module 4 of this information about protein structure, function, etc. So, in that same line I will be talking to you about protein fingerprinting, peptide mapping, and how different proteins can be purified, etc. So, this is module 4. I am Bishwajit Kundu again, those who have not listened to me earlier. So, to proceed, let me first talk to you about what we have been talking. So, we have something called hemoglobin and sickle cell hemoglobin that we have discussed in our previous classes. So, hemoglobin has 4 chains, 2 beta and 2 alpha. So, those are represented in different colors here. This is just a cartoon, and each one of these polypeptides or chains consists of one heme group, and each one of these heme groups can bind to iron, and each iron is capable of binding to oxygen, and therefore, you have oxygen carrying molecule hemoglobin, which is the main protein responsible for our blood being red in color and we being able to breathe oxygen and transfer the oxygen to tissues, where the carbon dioxide is exchanged with that of oxygen by this hemoglobin. So, if you look at the structure of these hemoglobin, because these are proteins and we have been discussing about proteins, then this is a normal hemoglobin, and if this normal hemoglobin gets packed into a normal kind of cell, then the cells would look more like a discoid or flattened discs, and therefore, those are normal red blood cells. But as I told you in the previous class, you have a different kind of this hemoglobin, where because of a mutation of glutamic acid right here to a valine, the hemoglobin A or the normal form of hemoglobin gets converted into HbS or sickle cell type of hemoglobin. So, here is the difference, you have glutamic acid versus valine. So, this difference actually results in the difference or alteration in the structure of this protein molecule, and the altered structure results when gets packed inside the cells, results in the cells altogether being altered. So, the structure of the cells gets altered, they become sickle like in shape. So, they are called sickle shaped, and because of this sickle shaped, they actually start coagulating and cause various kinds of problems. Now, when this was visualized, initially people would not know how this HbA, that is this type of cell and this type of cells, differ. Only thing that people would know initially was that there is a difference between these types of cells which are perfectly all right versus these types of cells which weren't all right. So, to look at the difference, initially what happened was, there was a very fantastic scientist called Vernon M. Ingram, the name is written here, you can see, and he was supported by Max Perutz in his lab, probably his student, and actually did a fantastic job to first time detect and decide why and how these amino acids do change in the proteins and what is the consequence of that. So, what he did was, he took 2 test tubes, and these 2 test tubes had hemoglobin normal one and the sickle cell hemoglobin. So, what he did was, these 2 hemoglobin test tubes were treated with trypsin. You remember in the previous class I was talking to you about trypsin. Trypsin is an enzyme, a proteolytic enzyme, that means it actually breaks the protein chain. So, you have a trypsin, you add trypsin to this, you add trypsin to that, and if I just want to remind you that trypsin actually breaks the protein after the positively charged amino acids. So, you have, let us say, the amino acid A, B, C, and the C amino acid is positively charged, which can be lysine or arginine, which are positively charged amino acids. So, immediately after the positively charged amino acids, the chain gets snapped, clicked. So, once this is clipped, so this is also clipped, this is also clipped. So, HBA, which is shown with a slight reddish color, and HBS, which is shown with this gray color. HBA is the normal one, HBS is the sickle celled type. So, what you can see is the alpha part of hemoglobin. Let me go back to my previous slide to show you that there are 2 alpha chains and 2 beta chains, just to remind that. So, here are the alpha chains shown in blue, here are the beta chains shown in red in one case and gray in another case. So, red suggests they are normal because we are looking at red blood cells and hemoglobin, and S suggests they are not, the gray suggests they are not normal. So, we are looking at sickle cells or sickle celled hemoglobin. What it means is that in hemoglobin A, that is normal type, as well as hemoglobin S, that is the abnormal type or sickle cell type, the alpha domain remains normal, but that was not known to anybody before this particular set of experiment was done, and that actually opened vista of information through which now we have a whole lot of information about proteins, and whatever I have been teaching or talking to you in these sections are actually a culmination of wide range of experiments that gradually started building up, and we get to know more and more about proteins. So, this is one such first set of experiments performed by Vernon M. Ingram. So, what he did, not knowing that there are differences in the chains and the chains would be like that and so on and so forth, what he did was he took normal hemoglobin and the sickle cell hemoglobin and put trypsin into it. What would have happened is this hemoglobin, whether it is alpha chain, beta chain, everything will get snap broken into small pieces. So, hemoglobin A would get into small pieces, hemoglobin B would also get into small pieces. Now, what you do? You just put one drop of those small pieced hemoglobins, that is a broken fragmented hemoglobin A, on a piece of paper, a paper which can actually transmit components along by simply the diffusion process or by mobility of the components in the paper. So, those are tissue papers or some kind of paper. So, if you have this paper, I am not disclosing it right now because there are many more, and then so you have this paper and you put a spot of the normal hemoglobin on that paper and apply electrical influence, that is charge of negative on one side and positive on another side. So, now as this hemoglobin A, which is broken by trypsin, consists of small fragments, each fragment depending on the overall charge of this fragment. You remember the amino acids, the peptides are made up of amino acid, and the amino acids may be charged. So, if they are charged, then depending on the full charge, the total charge on each one of these fragments will either move towards the positive end or towards the negative end. The positive end is shown with a small plus sign and blue in color, and the negative end shown with a negative red in color and a negative sign. So, depending upon the electrical charge on each one of these fragments, they will start moving towards the positive or the negative charges respectively. So, when you do that for both normal one as well as the sickle cell type, then each one of these small fragments will start moving differently to the positive and the negative termini. Now, what he did was, Vernon Ingram, what he did was, after he had run it in this direction horizontally, that is, this is negative charge, this is positive charge, and the small fragments depending on their entire charge or the full charge or the total charge, they have started moving to different directions, faster or slower or whatever, and they have reached to certain point. After moving for some time, you change the direction or orientation in this fashion, so that every point that has gone into discrimination on the basis of charges are now capable of moving in another direction to further discriminate the charge if there is any. And if you do so, that kind of chromatogram is chromatography. This process is called chromatography, and if you do it in 2 dimensions, that is, once in this direction, horizontal direction, and once is the vertical direction, that is called a two-dimensional electrophoresis. That is what is shown in this pictorial diagram here. So, in this cartoon what is shown is you have a paper and on which you have made a spot right at the center, and you have positive charge up there. So, you have connected it to a positive terminal of the battery and a negative terminal of the battery, and once you do so, you switch on, then what will happen, depending on what this spot consists of. In this case, the spot would consist of either HBA, which is fragmented, or HBV, HBS, which is also fragmented. So, these fragmented portions will actually move in the different directions depending upon the overall charge on these fragments. So, some will move towards the positive, some will move towards the negative. Now, once they have moved into different charge position, you turn it around 90 degrees. That means this was vertically going up, now you turn it around, just turn it like this, so that it becomes at the bottom, and then you start moving it in the opposite direction. That is, you put a positive charge here and a negative charge here, so that each spot is further discriminated. This spot will further move whatever components up and down. If you do so, at the end of it, what you find is, in the case of normal hemoglobin, you find certain spots like this, and in the case of sickle cell hemoglobin, not only did you find all the spots that are like this, but one of this spot, here you can see it in this place, actually has moved further up like that. What it shows is that this particular spot is different than the normal one, and here is the actual chromatogram that has been developed by Ingram, and this is available in the book called Stryer by Biochemistry. So, you see here is the origin, here is the origin, and this particular spot in one case called hemoglobin A is right here, and in this case it has moved further up. So, hemoglobin S has one particular spot which has moved further up or is different from that of hemoglobin A. So, what you have done? You have purified. So, pure hemoglobin and normal hemoglobin and SCHB is sickle cell hemoglobin are taken in separate test tubes, digested with trypsin, which cleaves after basic amino acids like lysine and arginine. Two separate strips of Whatman filter paper are spotted with normal hemoglobin and sickle cell hemoglobin, which now consisted of tryptic or trypsin mediated generation of peptides, and those were allowed to undergo what is called as paper electrophoresis at pH 2. So, the highly charged peptides will now migrate more towards the anode or the cathode, that is the positive or the negative charges respectively, and the paper strips are dried and then attached to larger squares of Whatman paper and chromatographed at right angles, as I just now mentioned. And once they are done so, it is not simply that after doing these chromatograms you will be able to see all these spots, but for that you have to do something else. The something else is that you have to dry them and stain them for visualization with some reagents like ninhydrin. The name is given here. So, once you just spray some ninhydrin on those filter paper, Whatman filter paper, then you will start some orange or yellow spots, just like what has been shown in this particular cartoon, appearing on the electrophoretic paper. So, what you do? The peptide map. Now, this is kind of a peptide map, or the previous one which I just now showed. This is the kind of a peptide map that gets generated with different colors. The peptide map of hemoglobin and the sickle cell hemoglobin are compared, and it was found that one peptide was differently placed, which I just now showed in my previous slide. So, one of these peptides is differently placed as compared to all the other dots or the spots. So, on examining this peptide and determining the amino acid sequence, Ingram found that it was a valine which is substituted for glutamic acid. Now, remember, glutamic acid is acidic. and valine is a hydrophobic amino acid, normal hydrophobic amino acid. So, if it is acidic, then what happens is, so the normal hemoglobin will produce a peptide with more negative charge, because the glutamic acid will have a glutamic acid group is COOH group, therefore, there is a COO negative charge. And if there is a highly negative charge amino acid in the fragmented portions, that negatively charged amino acid will allow the particular peptide which consists of that amino acid to move faster towards the positive charge, and as a result they will move ahead, as is shown in this case. So, that is what discriminates the two proteins, and that is how Ingram first came to know that there is a substitution of glutamic acid by valine. So, glutamic acid by valine. This single substitution of the valine for glutamic acid at the 6th position of the hemoglobin dramatically changes the structure of the sickle cell hemoglobin and make it make it from fibrous, make it to form fibers with the within the red blood cells, resulting in the deformation of the sickle cell, as is shown in this diagram. So, you see how beautifully this guy, how intelligently this guy Ingram first derived simply by doing some paper chromatography to decide on what are the amino acids and how we can actually generate the database, or what we can simply say, we can map each amino acid of a protein. This was the first time was shown by this guy. Immediately afterwards, people started knowing more about the proteins and peptides by another method called 2D or two-dimensional gel electrophoresis. So, the previous one was paper chromatography or paper electrophoresis. Nowadays, we do what is called as two-dimensional gel electrophoresis, and that technique is now very well known and is called peptide mapping. So, this was kind of a modification of the paper chromatography technique, paper electrophoretic technique, and this was developed by O'Farrell. So, peptide mapping, when I say peptide mapping, that means you are given a protein, you drink milk, so there consists of lots of proteins. You have an egg that consists of proteins, you have the pulses, the dal, everything has those proteins, but you do not know what these proteins are, what is the amino acid sequence, how to get that information. That is what each one of these peptide can be actually disseminated, the information can be obtained, that is what is called a peptide mapping. So, peptide mapping became a useful technique to compare similar proteins from different sources. So, for example, if you get proteins from milk and if you get proteins from dal or soya bean, what are the differences between these proteins? You can get the differences by doing this peptide mapping. And as I said, there are about 30,000 proteins at a time present in your cell. So, if you want to map all the 30,000 proteins, it will be a humongous task. So, what people have done slowly, we have actually come to know various proteins, thousands of them, and this database has kept on increasing. So, we can have a database of all the proteins that are available for a human cell, and that too, let us say you have kidney cells. So, all the proteins in a kidney cell, this peptide or the amino acid composition of those proteins. We can similarly have heart cells, we can have lungs cells, we can have various kinds of cells, we can have bacterial cells, we can have a plant cell, we can have any organism, mosquito, rat, monkey, so whatever you can talk about, we can actually take those cells and get the information of all the proteins present there and store them in the form of peptide maps, and those are the databases that we generate. And anytime you come across a new protein or somebody is talking about something new, or even if you wish to understand, so let us say what are the proteins that are so similar between me and a plant or me and an elephant, you can simply compare them by looking at the database. So, those databases are now being done, now being made available, and they can be easily compared by homology searches. So, what people do is, so the protein fingerprinting data, this is also called the peptide map, is also called a fingerprint map. Fingerprinting data has been further augmented with new database containing the 2D electrophoresis pattern. 2D is 2-dimensional electrophoresis pattern. So, what people do in 2-dimensional electrophoresis is that as compared to the previous diagram where Ingram actually used placed a spot on a paper which is shown here and then allowed it to move either in the positive or negative direction by allowing electrical potential difference between these two ends. So, they will start moving. In this case the same thing is done, only thing is that the paper is now replaced with that of a gel. So, what you do, you have a hollow tube, glass tube, and inside that glass tube you fill with something called a gel of polyacrylamide. So, this is a component, this is a chemical which when present, when you can actually dissolve it in water, and then after dissolving it in water you can let it polymerize with addition of some other components. But then as soon as you add those components and place it, it can be poured in a tube, but then if you keep it for a little bit time, then that will just polymerize and it will stay there. So, it will become a thick jelly like material. So, that is what is called a gel electrophoresis or gel. So, you have a tube in which you have the gel, and then you have something called ampholites. So, what are these ampholites? Ampholites are again some compounds which are differentially charged. So, some of them are very highly positive charge, some of them are slightly less positive charge or negative charge, and so on and so forth. So, you have a range of charged molecules. So, after you have created this tube which is shown here, you at the top of the tube layer just add a little bit of these ampholites, which is a mixture of all the different kinds of charged molecules, and then allow them to go through this tube through the gel material. If you apply electrical voltage across these two, then some of the molecules which are highly negatively charged or highly positively charged depending upon the polarity of the tube will start migrating towards the other end as is shown in this diagram, and then they will take places or positions in the tube corresponding to equivalent to their charges. So, you have for example, here the most positively charged molecule and here the most negatively charged molecule, or vice versa. You can have the most negatively charged molecule here and the most positively charged molecule here, and then intermediary molecules which are shown by different bands in this particular diagram. So, once that is done, this is one dimension, that is the first dimension. Now, they have actually migrated to different positions, but then you do not know whether this particular band here right here is actually consisting of only one kind of molecule of particular charge, or they do have same charge, but difference in their molecular weight. If you want to do the discrimination, then you just turn it around, that is place it in a different direction, turn it around and place it in a different direction and let it flow on another gel which is called SDS polyacrylamide gel. What it will do is, now each band depending upon its own constituents will start flowing or moving in the other direction, but here also you have a gel, and this gel discriminates on the basis of its mass or the size. The bigger the size, the slower it will migrate through the gel. The smaller the size, they will migrate faster. So, not only did you separate them on the basis of charge, now similarly charged molecules are again separated on the basis of their mass or molecular weight, and then you get a pattern. This pattern actually discriminates each one depending not only on their charge, but also on their mass, and therefore, you can get the information, the entire information of a polypeptide. This is what a 2D gel electrophoresis typically looks like. So, you have, as I said, you can have a gel inside a tube and then run it in one dimension. So, that is the first dimension. Followed by that you turn it around, just move it across like this, turn it around and put it in another gel, in which you start moving, separating them on the basis of its size, and once these two are done, you get a number of spots. So, the first dimension is called isoelectric focusing. Why it is called isoelectric focusing? This isoelectric focusing because these ampholites, as I told you, would migrate to a point where their electrical charge is balanced by the charge of the voltage that you have applied. So, wherever the charge is positive 1, 2, 3, 4, they will just migrate up to that point and then balance there. So, that is called iso means similar electric focusing. So, they get focused in the particular place. So, what actually you do is you run the ampholites first, so that they migrate and form a gradient all across, and then for example, if you have hemoglobin, you can cut the pieces of hemoglobin and put it on this gel right here on top of here. Then each one of those pieces would migrate only to that particular location where the charge of that piece is equivalent to charge of the ampholite that is already made a gradient from top to bottom, and those are the ones that are shown as these nice bands here, just for example. So, that is why it is called isoelectric focusing. Once you have done the isoelectric focusing, you can again move them depending upon their molecular weight. Mr represents molecular weight here. And just for giving you some excitement, here is the actual electrograph that was developed by Ingram. Well, look at this is 1961. This is called the fingerprints of hemoglobin. So, hemoglobin as a polypeptide has different amino acids, and the entire polypeptide can be broken into small pieces. Each piece can be separated based on their charge as well as their mass. So, that is 2D gel electrophoresis. Now, moving on, once we have the idea of 2D gel electrophoresis, before that the paper electrophoresis, and then we came to know about very important differences in amino acids based on their peptide composition. So, it was obvious that we should be able to separate out proteins from different components and start making more and more useful products. So, here are these spots are very different, that is what is shown with this diagram. So, we move ahead with the purification of proteins, because we need to have proteins in larger quantities. It is, as I told you right in my first class, that proteins are workhorses. We need to eat proteins, we need to supplement proteins. Proteins are enzymes that are utilized in various industries. Proteins can be therapeutic agents, can be utilized in the drug industry, and so on and so forth. So, proteins are everywhere, and therefore, we need to purify proteins. Where do you get these proteins from? Obviously, you can get the proteins from any organism, but then microbial or microbes or microorganisms are easy to get these proteins from. Plants and animals are also good sources of these proteins, but you cannot indiscriminately kill plants and get the proteins, or destroy plants and get the proteins, and so on. Similarly, for the animals, but then microbes can be grown in larger quantities in cultured vessels, and from there you can actually get the proteins. So, the steps that are involved in getting or isolating proteins from microbial culture, plants and animals, or various sources by the different separation techniques are collectively known as downstream processing. Now, which protein and what kind of technique, separation technique would you use, is something that depends upon these three parameters. What? The exact source material, whether it is plant, whether it is microbe, whether it is animal, whether it is virus, whether it is some yeast, whether it is fungus, whether it is lichen, whether it is so it depends upon the source of the material from where you want to have this protein, whether it is inside the cell intracellular, or it is produced and thrown outside the cell, it is extracellular. So, all these components, so exact source of the material or the protein decides the purification strategy or the downstream processing. Next, it depends upon the quantity of protein required. So, how much protein would you require? So, for example, in the paper and pulp industry, or in the leather industry, where you require huge quantities of these enzymes for treatment of the leather or the paper or the pulp. So, there you require very huge quantity, and therefore, the raw material has to be processed in huge quantities, and the requirement is very high. So, obviously, the downstream processing for those processes are going to be higher or different than anything else. And the third, which is much more important than any of the other two, equally important rather, are the physical, chemical and biological properties of the protein. So, every protein, as I said, folds and then takes up a special structure, therefore, it has a physical property, whether you can actually heat it, it can withstand certain temperature or pressure or pH is something that the physical properties or chemical properties actually determine. So, once you have taken care of these things, what are the different protein purification procedures available to us or downstream processes available to us? So, first of all, protein release. If you look at any cell, here I have taken an example of an animal cell. So, you can see there is a cell, there are these components, there are proteins inside the cell, and then there is a membrane which is shown here as a bimembrane, and then there are proteins that can be secreted outside the cell. There are some proteins which are transmembrane, that means they are either inside, half outside, mostly outside, some inside, and whatever it is. So, they are transmembrane, there are intracellular, they can be extracellular outside the cell. So, extracellular inside the cell, intracellular or transmembrane, whatever way. First of all, what you need to do is you have this whole gamut of proteins present in the cell. So, I am just drawing something from here, expanding it. So, if you do so, the first thing that you need to do is disrupt the cell membrane if they are present inside the cell or transmembrane. So, you got to break these cells open to get the proteins out. The first step in the process is actually you do something called laboratory scale disruption of the cell membrane. This can be done by either sonication, French press, enzymatic or chemical cleavage, or freeze thawing techniques, as has been listed here. So, what is sonication? Sonication is, you see, this is a machine which is a sonicator machine, and what you do is you have a probe, and this probe actually produces ultrasonic sound waves. So, if you dip this probe into a culture means a flask in which you have these kind of cells present in large numbers. So, that culture, if you dip the probe and then start the machine, it will start producing very high ultrasound sonic waves, and those waves will disrupt these membranes, that is, these membranes will be broken and the cells will die and all the components will just burst out. French press is a similar machine. Here sonication is not used, rather a very high pressure pressing method is used. So, all the cells are put in a very small area and very high pressure is applied, and as a result the cells will burst. Then you have enzymatic or chemical cleavage of the cell. So, as you see, these components of the cell membrane are also made up of lipids and some other proteins. So, you can have some enzymes which breaks these lipids and the proteins. So, those are the enzymatic and chemical cleavage. So, those will break up and the components of the cell can come out. And the final one is freeze thawing technique, where you rapidly freeze the cells and then rapidly heat the cells or thaw the cells. So, if you thaw and freeze and thaw and freeze, after some time these membranes will be broken. So, that is another technique. Apart from that, for larger scale, for example, you want to produce huge amounts of proteins, then you can have a high pressure homogenizer or bead milling. Homogenizers are also again high pressure machines which can provide much more pressure than the French press, which is shown above here. And then bead milling are something where you have the culture of the cells and then you put some glass beads inside the cell and then rotate it very fast, so that everything gets broken. So, that is bead milling. After you have broken the cells, all the components inside the cell as well as the cell, so all the proteins desirable, undesirable, membrane components, cellular components and other everything, just so whatever is shown here just comes into the medium. Now, what you do? You centrifuge. What does the centrifuge do? The heavy molecule, centrifuge means you take it in a tube and rotate it at it. So, the centrifugal force actually pulls the heavier molecules towards the outer end of the tube, and only a clear solution will remain at the top end of the tube. So, all the debris, all the components that are not required, or even if they are required, they are separated out in two phases. So, there will be some phases which have all the debris and some phases which are clear. So, you can easily separate out the components of the cell from those which are soluble components. Once that is done, then proteins can be purified according to their basic characters, such as solubility, size, charge and specific binding affinities. So, these are some basic characteristics of the protein. They can be very, very soluble, they may not be soluble. Their sizes may differ, as I told you in case of electrophoresis, they can be very large size, they can be smaller size, they can be multimers, they can be smaller monomers and so on. They can have different kinds of charges, highly positive, less positive and so on, depending upon the amino acid and their specific binding affinities, because the proteins do interact with each other and therefore, they have binding affinities. So, one protein might have binding affinity for another protein, or one protein might have binding affinity for a DNA, or one protein can have binding affinity for a lipid and vice versa. So, usually the protein mixtures are subjected to a series of separation procedures before we get a pure protein yield, and the first of this is called the salting out effect. All these pictures are very kindly actually taken from this Creative Biomart recombinant protein slide, which is present in the YouTube. Thank you very much for providing us with this information, which can be utilized for teaching purposes here. So, the first one is salting out. Now, what this salting out does is, I told you initially that the proteins do remain soluble, may remain soluble in water depending upon. So, if you have depending upon their charge. So, for example, if you have a protein like this, then there may be charged amino acids on the surface of this protein, which can interact with water by forming hydrogen bonding with water. So, more and more this thing is capable of forming hydrogen bonds, it can become soluble. Anything that gets soluble in water is because it can readily form hydrogen bonds with water. Now, if you start adding salt in a beaker, let us say we take a beaker which is shown here, not this beaker, but let us say in a test tube, and then there are various kinds of proteins as has been shown by these dots, colored dots, and then you start adding salt. Now, what will happen? If you keep on adding salt, initially the salt will keep dissolving, but gradually if you keep on adding more and more salt, the salt will start interacting and scavenging the water molecules that are present on the surface of these protein molecules. So, whatever was stuck to the surface of these whatever water molecule was stuck to the surface of this protein are now taken out by the salt itself, because the salt themselves want to solubilize first, they want to break their crystal lattice. So, if that happens, the protein will be naked of all the water molecules. So, I am a protein, this is another protein, I am scavenged, all my water molecules are being taken away, all my water molecules are taken away, water molecules from this are also taken away. So, if all these water molecules are taken away, then there is nothing in between this protein, and therefore, they will just clasp together, and that is what is called salting out precipitation. So, you will get a precipitate of the desired protein at different salt concentrations. Once you have done that, you can centrifuge and take out or throw the supernatant if that is not of your choice. So, if you are interested in this protein, you can take this protein, but then you have to get rid of the salt, because it is the salt that has created the lump of this protein to form. So, you can get rid of the salt by what is called as dialysis. So, what is dialysis? In dialysis, you take a membrane or dialysis bag, which is shown by this one, the yellow colored component, and then you put your protein inside, and this dialysis bag or the membrane has perforations, or this dialysis bag, so perforations are shown here, the perforations of different sizes. So, you can have a 30 kilodalton dialysis bag, a 100 kilodalton dialysis bag. What it means is the perforations will allow any molecule smaller than 30 kilodalton or 100 kilodalton, but bigger than those molecules will be retained by the dialysis bag inside the bag. And if you put them in a buffer or a solution or water, then what will happen is all the components which are smaller, for example, the salt which was initially able to precipitate this protein out, will now release this bag and the protein molecules will only remain. So, that is another step of protein purification. Moving on, you can have different chromatographic steps. The first one of them is easy to understand, is a gel filtration chromatography. Now, gel filtration chromatography separates proteins on the basis of their size. What happens is, in gel filtration chromatography, you have a tube like this, which is typically called a column, and this tube will consist of beads, small spheres, and these spheres are actually sponge like material. So, imagine that you have made spheres out of a sponge. So, these sponge balls are now put in a jar, as you might have seen various juice shops having those oranges in a jar. So, what you see is in between those oranges there are some gaps and holes, and so if you pour water it will just come out from the different sides. So, if you pour a sugar solution, that will also come out. If you pour a very small fine type of solution, that will also come out. But if you pour something which is slightly bigger, that will have to interact with these balls. And if you imagine that those are sponge balls, then there are perforations. So, even the smallest of the things that you have poured in, smallest of the components that you have poured in, let us say you have a mixture of these components which are blue dots, the red dots and the green dots. So, you have three sizes of molecules, the red size being the biggest, the green one slightly smaller and the blue one the smallest, and you want to separate these three components from each other. What you do is, these are the sponge balls with perforations. So, if you allow these to just flow through this tube which consists of these sponge balls, then obviously you will see that the largest balls, largest components, that is the red one, they cannot actually enter the perforations that are shown right here, and therefore, if they have to come out, they have to come out between or interstitial spaces between the perforations. So, if you have a sponge ball, the bigger molecule will actually go all around the sponge ball and they will come out, but a slightly smaller molecule may actually get stuck into one of the interstices of the sponge ball, because sponge already consists of these perforations, and therefore, they will get stuck here and there and then finally come out. But the smallest one would have to actually interact here and then go somewhere else and somewhere else. So, they will traverse a longer path in the same set of column molecules as has been shown here. So, what happens ultimately is that the biggest molecules will come out first from the tube, followed by a slightly bigger molecule or intermediate molecules which is green, and then the smallest molecule which has actually entered into all the interstices, probability of entering into all the interstices will be subjected to maximum resistance, and therefore, they will come out last. So, that is what has been shown here. So, if you just put a solution of a mixture, then the mixture can be easily separated based on their mass or size, the largest being the first to come out or elute, and the smallest one being the one to come out last. So, in that process you can actually have a very good separation as has been shown here, and you can keep on collecting those components in different tubes. So, when the first one is coming, you can start collecting it here, the second one coming start collecting here, and so on. Moving on, you can have some more different kinds of chromatography. One is called the ion exchange chromatography, which is shown here, and the third one is called affinity chromatography, which is shown here. So, what does ion exchange chromatography say? As I told you, a polypeptide is made up of amino acids and they fold in a typical three-dimensional structure, and then some of the amino acids will be at the surface, some of the amino acids will be at the core of this structure. But then depending on what are these amino acids which are at the surface, which are at the core, and the overall structure of the protein itself, the overall charge on a particular protein can vary, as has been shown in this particular diagram, where you have some positive charge and some negative charge. So, this is one protein, let us say it is in the shape of a sphere, and then there are some positive charge and some negative charge, but the overall the charge is, let us say, positive or negative or whatever. So, let us assume that it has some charges, and those charges are overall positive or negative. Now, in ion exchange chromatography, you can have two kinds of these beads. In the case of previous chromatography, I told you that these beads are sponge type of material where perforations are there within the beads. So, these are the beads where there are perforations. In this case also you have a column, and the column will have the beads, but the beads do not have any perforations, rather they have charges on their surface. And those charges can be defined by derivatizing these beads with carboxylate group like CM or carboxymethyl group, or DEAE group like diethyl aminoethyl group. Now, they will have different kind of charges. For example, in this particular case, the beads has been shown to have negative charge. Now, if you have a mixture of proteins which have different positive and negative charges, which is shown here, for example, the green one has more positive charges and the blue one has less positive charges. So, the more positive charge will interact or try to bind with the negatively charged beads like this more tightly than the ones that are less positive charge. Those which are negative charge will never bind, they will rather flow through the sides of the beads, because these are negative charge, these are also negative charge, they will repel each other and therefore they will never bind. So, depending upon a mixture of different protein molecules, the surface of the protein molecule having different charges, those which are absolutely negatively charged, they will never bind to these beads and they will come out fast. Those which have slightly more positive charge will bind at slightly higher affinity exchange capacity, but then finally those which have the highest positive charge, they will keep on clinging to these beads tightly. Now, once that has happened, you have gotten rid of most of the negatively charged molecules, but still you have some of the positively charged molecules which are bound differentially to the beads. So, highest positive charge are bound somewhere, lowest positive charge are bound somewhere, but they are still bound. So, what do? You now elute the charged molecules with a salt solution like NaCl. Now, what does this NaCl do? You remember, NaCl actually is made up of Na plus and Cl negative ions. So, once you add the Na plus and Cl negative ions, these Cl negative ions, the Na plus ions and the Cl negative ions, depending upon what their affinity is, they will come and start binding to the beads. So, which one will bind to the beads? Yes, obviously the Na plus ions will bind to the negatively charged beads, and if they start binding, remember those are very small molecules. So, they can come, and these are small ions. So, they can start coming and binding all around, and they essentially replace these positively charged or feebly positively charged ions, protein molecules, and those protein molecules will be eluted out. So, simply adding NaCl will result in elution of the protein molecules which were bound to the beads. So, that is why what you have essentially done is one ion, which is the protein molecule, is now being exchanged for another ion, which is the Na plus molecule. That is why this is called ion exchange chromatography. The third type of chromatography is called affinity chromatography, and as I told you earlier that proteins might have affinity or likeliness for another protein. Proteins might have likeliness or affinity towards a DNA molecule or any other chemical entity. Drug molecules have affinity towards some proteins, and that is why they are drugs. So, that particular property of proteins which have affinity or attraction towards another molecule have been utilized very nicely. That is what is called affinity chromatography. So, for example, if you have a set of proteins where the mixture of proteins which are shown here as red, blue and green dots. Now, some of these proteins, let us say the red ones, have special affinity for these beads. The affinity is simply because these proteins have histidines, which is shown here, and the histidines have affinity towards nickel ion. So, what people have done is they have made these beads which are shown in the gray colors here, just like these beads, but in this case this was charged. In the previous case it was perforations. In the case of affinity chromatography, these have been derivatized with the presence of a nickel ion. So, they have been made in such a fashion that there is a bead which is shown here, and the bead consists of a long arm, and the end of the arm actually have a nickel ion. Now, this nickel is capable of attracting, or in a reverse way we can say that a protein having histidines get attracted towards nickel, or what we call as this protein have affinity towards these beads. So, when you add a mixture of these proteins and the red one, assuming that has this histidine, will go and bind to the beads, because the beads are exposing or showing them, look, I have the nickel, come on, bind to me. So, they go and bind, and in the process what happens, rest of the molecules which do not have the histidines, they will just come flow through, and then you can simply add, as in this case, a high salt buffer and let the proteins which were bound to this nickel elute. So, this way you can purify the protein. Now, I have talked to you about the different chromatographic procedures. I have also talked to you about different electrophoretic procedures. This is the last one where the fractions of each one of these procedures are shown on a gel electrophoretic pattern. So, for example, if you have the cell, you broke the cell and then the entire cell component is called the homogenate. You separate the homogenate on the basis of their size on a gel, and you can see each band represent one protein or multiple proteins of similar molecular weight, and you can see there are so many bands from one cell or a few cells of the same type. That is the first step of purification, that is why it is numbered 1. If you go for the next step of purification, that is fractionation, which can happen by salt or what is called as salting out effect. If you try to remember each one of those, I will just quickly take you through the processes. So, you have salting out, that is the first. So, first you break the cells, which is shown here, so that is called homogenate. Then you go for salting out and dialysis, followed by different kinds of chromatography, ion exchange, affinity, and then here it is the final stage. So, what you see is the first step, which is homogenate, has so many bands, you just cannot figure out which one is your protein. You go for fractionation, that is by ammonium sulfate precipitation or salt precipitation. If you do so, still you get so many bands, although some of the bands have thickened, thinned, and some have disappeared. Then you go for an ion exchange purification, and then you see some more bands have disappeared because those could not bind to the ion exchange column. Then you go for molecular exclusion chromatography or size exclusion chromatography, and then you get rid of many other bands and then concentrate on one band, and finally by affinity chromatography you get one band of your choice or the protein that you wish to actually purify. So, as we progress through the steps of purification, you can get a single band. That means you have actually found your protein of interest from so needle in a haystack, so many different proteins. So, that is how the purification procedure goes on. Now, I shift gears and take you to some very interesting and classical examples of what happens all around. So, in spite of the large biodiversity of microbes, the microorganisms, different kinds of microorganisms, viruses, we are restricted to certain bacteria or organisms which can be used as a source of protein. We do not use all the bacteria or all the organisms as a source of protein. We use only some of the bacteria or some of the sources. Why? Because many bacteria are around there, right there, which can be very, very harmful. We do not want those bacteria to be our sources of protein. We would rather use those bacteria which are very useful as sources of protein, and therefore, through our long history of understanding about microorganisms or working with microorganisms, we have now come across what is called as microorganisms that are generally recognized as safe, or in short it is called GRAS, GRAS. So, we have a list of these GRAS organisms generally recognized as safe organisms. One of the very common organism or microorganism is the E. coli, Escherichia coli. It remains in our own colon in the gut, and that is why it is called E. coli, Escherichia coli. So, these are non-toxic and generally they do not produce any antibiotics, and therefore, they are safe. Similarly, there are many plant tissues from which we can derive enzymes which have applications in the food industry, such as, and they must be obtained from non-toxic edible plant species. And one of the very classical example is that of a papain, which is obtained from this papaya tree. All of you know this. So, this papain is an enzyme. It is a very fantastic enzyme, and it finds its application in meat tenderization, clarification of beverages, as digestive aid, wound healing. So, various solutions, various pharmaceutical applications have been made, formulations have been made by use of papain. If you move on to some animal enzymes or proteins, insulin is a classical example. It is a peptide hormone, and I have given this example earlier also. Even up to 1980s, yeah, this insulin, and many of you probably at your home would have elderly people who are suffering from diabetes, and they would require insulin injection or tablets or something, or some kind of sugar test, sugar check. So, this insulin up to 1980s were actually produced from the pancreas, or obtained from the pancreas of cows and pigs. These cows and pigs had to be slaughtered, their pancreas were to be taken out, and then from there through all these chromatographic and separation techniques, you finally get some amount of insulin. But in the year 2020 itself, you know how many diabetic people are there now? There are about 20 million diabetic people. How can you just sacrifice so many animals to get so many pancreas and isolate such huge amount of insulin that can actually help the 20 million diabetic people in India alone? Fortunately, we need not have to worry anymore, because we have the genetic engineering processes which are in place, and therefore, we have genetically modified these organisms called the microorganisms, which are GRAS, generally recognized as safe microorganisms. The GRAS are like E. coli, and then we have made transgenic animals by direct microinjection of the DNA into the ovum of stem cells and produced insulin from their milk. And this technology is now called molecular farming. That is, it helps in producing pharmaceuticals using genetically modified plants and animals. The advantages of producing recombinant proteins in milk are: the productivity is high, the ease of the source material collection, you just milk the cow, you get the protein, moderate capital instruments required, and low operational cost, because you just rear the animals and the animals will give you the protein, and there is ease of production including purification and scale up. So, with that we have got a vivid idea about how the proteins can be separated, where the proteins can be obtained, and what are the genetically modified organisms that are present here at this point of time. So, with that I will end this session here and come back to all of you in the next session. Before moving ahead, I would like to acknowledge all those who have contributed. I have taken these slides, many pictures from different web sources. I thank all of them who have contributed and allowed us to have these here on the web sources from which I have taken and has been utilized very effectively for teaching and education purpose. Thank you very much.